In Part A of our virtual reality, we will be exploring a simple molecule, dichloro-difluoromethane, or CCl2F2. The first thing we want you to do is draw the Lewis structure of this molecule using perspective bonds and label the carbon-fluorine and carbon-chlorine bond lengths, and then 3 bond angles, the chlorine-carbon-chlorine, fluorine-carbon-fluorine, and then fluorine-carbon-chlorine bond angles that you determined in Part 2 using Chem Draw 3D. To begin viewing molecules, click on the green plus icon in the entry list window. Then click on the Search menu option on the left side of the Load window. Click in the Enter keyword box to enter information for molecules to view. For most molecules, we've provided you a numerical code to facilitate the exact molecule we want you to examine. So enter the code for dichloro-difluoromethane 6391 and select Load. If you see a window saying the trial period has ended, click the red X to close that window. Once the molecule loads, here are the handset moves that will allow for you to manipulate and examine your structure. Students will be asked to try them themselves and make sure that their instructor or TA has checked off that they're able to do each task. The first task is to use one grip finger on either finger to grab your molecule and move it around your workspace. Next, rotate that molecule while grabbing it to see it in 3 dimensions. Next, grab the molecule with both grip fingers, and while holding the molecule, move your hands apart to make the molecule larger. Now reverse that move. Grab the parts of the molecule that are furthest apart and move your hands together to make the molecule smaller. On your right-hand controller, press and hold the B button until you see the menu wheel appear around your wrist. Select Measure by pointing the laser line at that option and click the right trigger finger. Use the joystick to toggle between the different measure options. You will be using distance and angle. Go ahead and try to measure the indicated distances or angles, and again, TAs or instructors will be checking to make sure that they're able to do each task. To measure the distance between 2 atoms, make sure your watch menu says distance. Scroll over the first atom, you'll see it highlight, and use the trigger finger on your right hand to select this atom. Move on to the next atom and select it in the same way. The distance between atoms will appear above. Choose to examine one of the bond lengths from your Lewis structure on the previous page and comment on how it compares. To measure the angle between 3 atoms, make sure that your watch menu says Angle. Scroll over the first atom, you'll see it highlight, and use the trigger finger on your right hand to select this atom. Make sure your central atom is the second one that you select and move on to the third atom to complete the selection. The angle will appear above. Choose to examine one of the bond angles from your Lewis structure on the previous page and comment on how it compares. Next, we'll have you explore a small biological molecule, glucose, as compared to cyclohexane. We can only examine a few molecules at a time, so before starting this next exercise, delete your previous molecules. To do so, we need to get to the entry list window. You may need to X out of or move, you can grip, finger, grab, and draw the load window. Select each molecule that you wish to delete and select the trash can in the bottom right corner to clear out your selections. Make sure to select that you want to delete entries and not atoms. Enter the code for glucose 5793 and select Load. Enter the code for cyclohexane 8078 and select Load. Manipulate each structure, changing size, orientation, for example, so that they can be viewed side by side with each other. You must complete this step before answering the following question and having your instructor initial this work. Show your structures to your partner and then your instructor and rationalize if the structures shown are in a chair or a boat conformation. Lastly, we're going to explore a large biological molecule. We're going to look at the protein hemoglobin, the oxygen-binding protein from red blood cells. To examine the structure of protein hemoglobin, begin by deleting any previous molecules to start with a clean viewing window. From the Load menu, choose Source from the menu options on the left, and in the Search ID window, enter the code for hemoglobin 2DHB and select Load. This structure is significantly larger than those we've previously played with. Manipulate the size of your structure such that you can realistically hold it in the palm of your hand. Show your instructor this small structure. Now make the structure so large and physically pull it towards you so that you are literally standing in the middle of it. Show your instructor you hanging out inside of the structure. The structure of the protein part of hemoglobin is shown in ribbons mode. This is represented by 3 types of structures. Find each one and be able to point them out to your instructor. Alpha helices are shown in red as coil structures. Beta strands would also be shown in red, but as flat arrows. Do you see any of these? Loop domains are shown in green and generally connect helix and strand domains in proteins. Try tracing the ribbon from start to end. Do you only see one ribbon in this structure? What do you think this might mean? Hemoglobin is able to bind and transport oxygen because it binds a small molecule called a ligand. The specific ligand for hemoglobin is called heme. The structure of heme is shown in what is called ball and stick model, where each atom is represented as a colored sphere. See if you can locate the heme ligand in the protein and try and complete the following for the color typically used to represent each atom. Note, hydrogen atoms are typically not shown in large structures, but these would typically be white. Do you see the carbon atoms? Do you see the nitrogen atoms? Do you see the oxygen atoms? There is one colored atom, orange, that is at the center of the heme that is a metal ion. You may need to use Google a little for this question, or you just might know. But what is the identity of the metal that is bound at the center of the heme? A few parts of the protein are also shown in ball and stick model. These are amino acid side chains. One of them is particularly close to this metal atom listed above. See if you can figure out what amino acid side chain it is using the handouts on amino acids. Go in and measure the distance between the metal atom and the closest atom from this side chain.